Welcome all, and thank you for joining today's AWRI webinar. My name is Robin Dixon and I'm a Senior Viticulturist at the AWRI. Today I'm joining you from Erbrae on Kaurna land. And in this session, we'll look at nitrogen use efficiency in vineyards. In the spirit of reconciliation, the AWRI acknowledges the traditional custodians of country throughout Australia and their connections to land, sea, and community. We pay our respect to their elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. I would also like to acknowledge One Australia for providing funding and support for webinars via the AWRI Extension Project. So before we jump in, as always, a couple of quick reminders to anyone who's new to the AWRI webinars. If you would like to provide a comment or ask a question at any time throughout the presentation, just jump on the Q&A area on the Zoom toolbar down the bottom. You can type in your question at any time, and we'll be holding the Q&A session at the end. Also, this webinar, as always, is being recorded, and a copy of the recording will be emailed out to the participants shortly after the webinar. We would also like to collect some data today, trying to understand your knowledge on the topic. So you'll see a poll that has just popped up asking you two simple questions. So if you can please fill out the poll. The poll will also pop up at the end of Michael's presentation today, just before the Q&A session, asking you how your knowledge has improved after the presentation. So for anyone who has just joined, welcome. Today's webinar topic is nitrogen use efficiency in vineyards, and it's a great pleasure to welcome Dr. Michael Treby. Dr. Michael Treby is an agricultural scientist with 40 years of research experience in perennial cropping systems, working with CSIRO, New South Wales DPI, and AgVic. He has a strong interest in plant mineral nutrition as it relates to grape production in the warm irrigated regions of southeastern Australia. So Michael, if you're ready to start, I'll hand over to you.
Thanks, Robin. I'd just also like to acknowledge the traditional custodians of the land where I am, which are the Latchi Latchi people in northwest Victoria, but also just across the river, the Barker people who live up along the Darling and the lower reaches and the Murray down here more locally. Okay, right, as Robin said, I'll be talking about nitrogen use efficiency and trying to tie together a number of streams of science that have new knowledge that was developed both locally and by our colleagues overseas and or just generally elsewhere around the world working on this more general area. I intend to just use some fairly practical, to suggest some fairly practical solutions to making nitrogen use efficiency in vineyards to improve the use efficiency in vineyards. But let's try and remember we're dealing with an agricultural situation, and the expression, it's something very agricultural means just that. It can be a bit rough from time to time. There is no precise formula for just about anything really, but in relation to fertiliser management on vineyards, or indeed fertiliser management anywhere, on any production system, things, stuff happens. It's the real world. Oops. Okay. What I'll talk about, I'll just talk about needs versus fertility. So by fertility, we mean, what's available in the soil. Synthetic versus organic fertilizers, because that kind of thing, that comparison, that division occupies a fair amount of space in people's minds, some very emotional, philosophical, almost religious And it's not for us to make any value judgments about that. That's just a fact of life. People have some preferences in this matter. I'll talk about the fate of nitrogen in soils, and what that might mean for nitrogen use efficiency. I'll talk about uptake. So we're talking about vines taking up that nitrogen to do whatever it is we want it to do. And I'll talk about the fate of those grapevines, what happens to that nitrogen in those vines in relation to what we want to achieve. Trying to keep things in perspective, we've got to remember that out of every tonne of grapes, about 120 kilos of carbon and about 2 kilos of nitrogen are removed. And then I've listed all there the other major nutrients that are removed as well. We've got to remember that this is a finite system. Very early in the piece up here in the northwest of Victoria and southwest New South Wales, they mined the minerals very quickly. And within a few seasons, there were yield declines and bigger declines as they basically ripped out of the soil all the available, NP, K, and calcium, and so forth, and became very obvious very early in the piece that they needed to be replacing it. The situation with nitrogen is not quite as dire as that, because there's actually quite a lot floating around, and I'll show you some data just so you can wrap your heads around just what sort of numbers we're dealing with here. It's in some sense it's a bit of an accountant's sort of approach, but it's good to remember just what it actually takes to drive the system. Okay, now nitrogen inputs. This is kind of complicated, but we'll work our way through it. On the left, we've got dinitrogen. That's what's atmospheric nitrogen. Most of the atmosphere is actually nitrogen, dinitrogen gas. Some of that nitrogen can end up in organic matter, organic nitrogen in the soil. That's the arrow that goes right down to the bottom and across to the right. That's organic N, and that's in process what we generally known as fixation. And that's in leguminous cover crops, for example. Also by just free-living nitrogen bacteria, and they all produce organic N. Now, across the top, this is where we tends to be a little bit of debate. Some of that nitrogen gas can be fixed in what's known as the Haber-Bosch process into urea and ammonium-containing fertilizers. That process takes energy, and it also appears to divide people more or less into the conventional approach versus the organic approach, and for various reasons, they're opposed to that. That's their decision. The process takes a lot of energy, though, and that's another contentious issue, because fossil fuels needed to be consumed to produce fertilizer. But it made a lot of fertilizer available very cheaply quite early in the piece, at that time, when the process was discovered, when it was discovered, we were digging up a lot of bird poop, and we're using a lot of other animal waste material to try and meet plant nitrogen needs in general. Okay, we can just divide the fertilizers into two. Nitrogen-containing fertilizer, which we talk, potassium nitrate, calcium nitrate. They're the ones we dig out of the soil, and there's also ammonium nitrate I've included in there, but that's actually produced by artificial means. The ammonium-containing fertilizers, monoammonium phosphate, diammonium, ammonium sulfate. There are good reasons to use those on occasion, but as a general rule, we try and steer clear of them. The other big input is urea. Now, these two nitrogen sources end up as either nitrate in the soil or as ammonium, and we have various ways of losing them. The biggest thing with nitrate is just leaching. We rinse it past the root zone. The other process that is a bit of a contributor to the greenhouse gas production is denitrification. So that produces nitrogen oxide and nitrous oxide, and I'll talk a little bit about that in a minute. With ammonium, we can actually lose that as gas, and you can actually smell ammonia. it's not that hard to avoid, but it does happen. That process is called volatilization. With organic nitrogen, we feed into that via the fixation process. This is talking about that pool down the bottom, and via organic amendments, so plant residues, animal waste, composts of various kinds. I've also included in there foliar sprays. It's not actually an organic amendment, but it's another way of supplementing plant nitrogen needs, although some plants are more suited to this than others. And with vines, I'm not saying that the jury's still out. The table grape industry uses that approach, fairly religiously. But what I am saying is I've not seen really solid evidence that it works. But plenty of people swear by it, and well, that's the way it is. Now, the leaching, the parts we really want to avoid, I've just highlighted in red, crudely, I might say. The gases that were lost through denitrification, nitrogen oxide, nitrogen, and nitrous oxide, there are actually a few other little ones, but they're known collectively as nitrous oxides, or NOx. And the movers and the groovers in the climate change space get fairly excited by these. Collectively, we're not a great contributor. Viticulture in Australia is not a great contributor to that, but they do contribute. They're very powerful greenhouse gases, and so we do contribute to that overall. I suppose the climate change people would say that, well, we all contribute only a little bit, and if we all stop a little bit, it'd probably have quite an effect. I'd say though that those processes occur. It's difficult to control them. We can do things about them though, to minimise them, to try and just reduce the economic loss. When we lose nitrous oxides to the atmosphere, okay, it has that effect on the climate, whether it's marginal or not. It's not the argument, but the argument is really we have lost money. We've put stuff in and we've lost it. With the leaching, we try to minimise the amount down that we push out the bottom. Past the root zone, and that actually forms a bit of a pollution problem, goes into groundwaters and aquifers and so forth. So that's not a particularly good thing. With ammonia going into the atmosphere, just absolute dead loss. But I've been on orchards in China where when you walked in, all you could smell was ammonia, and it's fairly significant indeed. Okay, let's start talking about the numbers that are out there. We're talking about, here's this a slide here is about, I can't see it because, I'll try and close that. Yep, thank you. Soil N's quite variable, but nitrate is really extremely so. Now, nitrate's the main form of nitrogen the vine roots will be taking up. Main form. They will take up ammonium, but their preference is nitrate. Now, these are some local data, fairly organic matters in the soils, low by Australian standards probably, or not compared to say the Simpson Desert, but by international standards it's pretty low. So percent organic matters, about 1.2% up in the top 18, 20 centimetres, all the way down to about 0.26, down 1.5, metre to 1.5. And if you like, about half of that organic matter is carbon. So if you just try and remember those sort of numbers, it's sort of helpful. The total amount of nitrogen in the soil is in the next column. So, it goes from about 770 up at the top, all the way down to 236. And if we just square two columns across, nitrate N goes from about 16.2 to about 5.3. So less than a third. Now, the really important point here is held in those two columns, which is called percent COV, percent coefficient of variation. And it being a biological system, a soil, those sort of coefficients of variation of about 20% mean it's really quite variable. If this was a manufacturing process and you had a coefficient of variation of that level, it means you're producing just absolute garbage. When we get to nitrate, though, the percent of coefficient of variation is about 80% at the top, all the way down to about 60%. But at some points, we measured a coefficient of variation of about 97%. What that meant was that if you took a sample in one spot and took a sample about 2 inches away, you get 8.5 in one, and you could get either 0 or 17 in the other one. That's what that means. So it's a hit and miss sort of thing. It's really quite variable. So what that means is, well, A, there's not very much there. And anybody trying to sample for that makes life pretty difficult. It means that it's, again, hit and miss to get a really accurate picture. Now, we start doing a nitrogen budget based on those sort of data. Total amount of nitrogen, about 0.05%. So it's about 500 milligrams of nitrogen per kilogram of soil. Now, all we're trying to do here is just get an idea in our heads of just how much is floating around the system. And across a hectare down to a metre is about 13,000 tonnes of soil. That means there's about 6.5 tonnes. That's 6,500 kilos of nitrogen total. Now, compare that to what your nitrogen inputs might be on your vineyards at the moment. And you think, oh, that's chocks there. Why are we worried about it? Well, we're worried about it because most of the nitrogen that's in the soil is actually organic. As I showed you, there's all that nitrogen, the total amount there, and then how much nitrate was there, which is the mineral N component. That gets mineralized. And from estimates we've made locally, we think that there's about 0.4 to about 0.9% of the total amount of N produced by that soil per year varies between 26 and 60 or 59 kilograms per hectare per year. That's across a whole year. Now, that's irrespective of when we want those vines to get it. That's irrespective. So it may or may not coincide with vine needs. Okay? May or may not. Okay, so now we know how much is floating around. Now we know potentially what our soils might produce, and it could be different all over the place. It will be influenced by things like wetting and drying events. And by that, I mean, well, irrigation for a start, but rain equally. And it'll be influenced by things like cultivation. Cultivation will cause a flush of organic matter decomposition by bugs. And in that process, they will mineralize in. Okay, now, for many years, we've cover cropped with the idea that we would be producing organic nitrogen that would then feed vine nitrogen needs over the coming production, or over the coming season. Yes and no is the answer to that. We've cover cropped up in this region since probably the mid-20s, early 30s, and it was done religiously. Faber beans formed quite an important component. That's about the 5th line down there. But when they were worked in by disking, and more recently by rotary hoeing and whatever, they were generally gone. The plant residues were just gone in about 3 weeks. So these things break down, and they do break down. Some will break down quite quickly. And that's the point of this particular exercise, of this particular slide here. So I've listed a lot of cover crop types: cereals, pulses, medics, brassicas, mixtures, and then grapevine material, because we're leaving material in the vineyard. Prunings, leaves, and so forth. They have what we call a C to N ratio. That's just a ratio basically of the percent C versus percent nitrogen. And some of these cereals have quite high levels, and high doesn't necessarily mean good. Cereals, for example, wheat's about 52, and then all the way down to triticale is about 41. Now, the significance of these numbers is that the higher that ratio, the longer that material takes to break down. So you get all the way down to grapevine material. So 3-year-old wood is a sultana, 3-year-old wood, 97. That's why that material will stay in your vineyard on the floor for quite a few years. It doesn't break down quickly. A more accurate measure of how quickly something will break down, and any of these materials will break down, is the percent in extractable polyphenols plus lignin divided by the percent nitrogen. And that's what I've listed in that third column. And the different coloured numbers mean that will refer to sort of categories on the next slide. But basically, the lower those numbers, the more likely it is that those cover crop material will break down reasonably rapidly. So when you see faba beans at 3.8, they break down quickly. Sandplain lupins, 2.4, break down quickly. Cassava medic, 1.2, break down quickly. All the way up to the vine prunings, grapevine material, so 14.9, all the way through to 31.2. But perennial ryegrass and wheat equally break down pretty slowly. Now, I'm not trying to put, I'm not trying to cast a value judgment on the use of any particular cover crop. It's just in relation to trying to supply vine N needs, trying to coincide or coordinate the breakdown of that cover crop in relation to when that vine is looking for it is pretty hard. But there are an enormous number of quite justifiable reasons why a cover crop should be grown or could be grown between vine rows. Organic matter is obviously one of them. But there's plenty of others there. And then my understanding is that there'll be somebody within this program of webinars who will speak about that. And indeed, there's been a long history of this kind of, or looking at that, the contribution of cover crops to soil health up in this part of the world. Remember, we have very hot summers and lots and lots of wetting and drying cycles. So the depletion of organic matter is significant. Okay, now I use those color codes in the other thing just to demonstrate, to indicate, the relative speed with which these things will break down. Those type 3 that I spoke about, those green ones, that included medic, faba beans. As I said, they break down really, really, really quickly. The next type, which had a sort of intermediate ratio of that those couple of components versus percent N, they take longer to break down. The other ones, there's a net immobilization, and they actually will draw nitrogen, mineral nitrogen, out of the system in order for bugs to access the carbohydrates in order to meet their energy needs. And that's a phenomenon known as nitrogen drawdown. And you can see, for example, when you put oats around young roots, around young vines, oats will actually drag nitrogen out of the system, and those vines can be the growth of the establishment of those vines can be retarded as a result. So, like I said, it's coinciding the breakdown of those cover crops and their composition, of course, with vine nitrogen needs, and that's a little bit more difficult to do. It sounds like a really great proposition, and it probably is, but it's hard. Okay, now get down to some harder core sort of things. Proposed in 1988. Two cycles, vegetative and reproductive, and we need to, well, what I'm raising here is just that we need to manipulate the supply of nitrogen to coincide with particular things we want to manipulate. And the really big ticket items we want to manipulate are inflorescence initiation and development, and then berry growth and ripening. Now, we also want to manipulate shoot development, so shoot growth and cane maturation, and then we would like also to manipulate how well those vines go into dormancy, and then how well they swing out of the period we're in right now, which is eco-dormancy, into auxiliary bud development and bud burst and so forth. Now, we need to remember that these things, it's a multi-season cycle. And what we do last year what we did last season, sorry, will have some impact on how we come out, how those vines come out of dormancy and set up those canopies to produce that crop, grow and produce that crop that's in the buds now. What we do this coming season will influence how much initiation and development, so how fruitful those buds will be the subsequent season. And that's really, really critical. Now, there are a number of other processes going on at the same time as all of that. You know, often we can't actually see all that, which I just try to highlight here. For many of us, in many parts of Australia, the bleeding process is going on. That's when sap flow starts happening. But there comes a period in late spring, early summer, where we're talking about there's a lot of new roots development, new roots being developed and emerging. And that sort of tapers off over summer, and then it tapers, and then it arcs up again as we go into autumn, more or less, round about harvest, more or less, and then tapering off again. That is really, these two periods are really critical, and leads me into the next slide. I realise all that's complicated, and it's also tempting to think, oh, that's just a really discrete period of the production cycle, and that only happens in that period. No, it's probably going on more or less most of the time, but the really big flushes or the really big activity periods are more or less described in that diagram. And so if we're looking to try and get more bang for our buck in a whole range of things, we'd be looking to try and coincide what we do with the various those various flushes of whatever it is we're trying to manipulate. Inflorescence, initiation and development, for example, is really, you know, that sets us up, really. Okay, now we'll just simplify that to go into this. We can talk about three distinct things that are happening in the vine. So we're talking about periods of major nitrogen uptake and internal movement. So mobilization, so that's the nitrogen that will be mobilized from the perennial parts of the vine to support spring growth. We have two periods of major mobilization, in spring and then in sort of about a third of the way through summer into early autumn. And that coincides with growing those berries and filling them up. Okay, now uptake really kicks off in late spring. So we're talking about sort of mid-November, you know, October, that kind of period. And that coincides, of course, with bud initiation. So, and the linkage between how well fed those vines are with respect to nitrogen and their bud fertility is well established, both locally here and elsewhere. And when we say that, we're not talking about trying to maximize that bud fertility. We're talking about producing, getting it to be at a reasonable level. Let's remember, you can take bunches off a vine, but you can't put them on. And if bud fertility is low or limited by vine nutrition, well, it's a bit late. Can't do anything about it. It could also be limited by the season. If it's really cold, and there's not much we can do about that, but nor can we predict if it's going to be so cold that it's going to affect it. So two periods of uptake. And then I spoke a little bit about the storage. I mentioned storage. Storage is happening when the crop's gone, leaves are exporting nitrogen into the permanent parts of the vine. So the trunk, the cordons, the canes, the roots. That's the nitrogen that's going to be mobilized in the following spring. So so up to now we've talked about the numbers that's floating around the system, and now we've talked about what the dynamics might be, and now let's try and start pulling some of it together. Okay, just to emphasise, some of that nitrogen, most of the nitrogen that's actually going in, taken up during summer, is ending up in the grapes. About half of it is ending up in the grapes. We'll leave the vineyard. The rest is ending up in the shoots and the wood. So that tells us something about the fate, and it sort of supports that contention, the idea I raised, that we can manipulate it during particular periods of the year, particular periods of the production cycle. And that's pretty critical. There's also another, and I did mention what we do one season affects what we do in another. So these are local data. Two seasons, long time ago, 1997, 98, 98, 99. In the first year we supplied, so that's the first year is the graphs on the left-hand side. So those three graphs on the left-hand side. We supplied 0 versus 100 kilograms of nitrogen. Now, remember what I said about how much was floating around the system, 6.5 tonnes, more or less. We're supplying about 100, and that'd be the outer limit for most wine grape production systems. Wouldn't be the outer limit for table grapes, for example, nor dried fruit too, for that matter. But for wine grapes, it's probably over the top a bit. Now, in the first season, we couldn't tell about how any difference between the amount of nitrogen we found in the vine, in the shoots, the leaves, the perennial parts, or the total amount, despite some getting 0 and the rest getting 100. In the following season, all those vines that have been supplied with 100 units of N just took off straight away. The growth was better, and the amount of nitrogen floating around the system was greater. And that was reflected in how much nitrogen ended up in the fruit, and hence how much was removed, and then how much those vines, when they went to sleep, how much extra they had in the perennial parts. Now, there's two things I want to get out of here. One thing that's obvious, on the right-hand column is just how many kilograms of nitrogen per hectare the grams of nitrogen per vine mean. When we look at how much those vines went to sleep with, so that's the middle bar, they went to sleep with about 100 to about 125 kilograms of nitrogen per hectare in the perennial parts. And remember, our inputs are likely to be around about 50, 60, maybe 70 odd in a wine grape production system. So from year to year, the vines that supplied with 0, they've still got about 100 units of N in the perennial parts. The amount they exported, though, was pretty small, and the crops on them also reflected that rather sad fact. So so there's about 100 units, and really our annual application is really just putting in enough to keep that cycle happening. The other thing I'd like to draw your attention to is the difference between the second year, the vines that have been fertilised and the vines that hadn't been fertilised. There's only about 20 or 30 odd units of N difference in the perennial parts. That puzzled us a bit until we went back and analysed and looked at the trunk. And really, it's only the outer third of the nitrogen, in the outer third of the trunk, that the nitrogen's actually mobilised. So very small amounts were quite significant to, in terms of when you just looked at the outer third of the trunk. Very small amounts in total were actually quite significant when it came to, you looked at how much, where that nitrogen has actually been mobilised from. So the point is, what we did the last season affected what we did this season, and what we did last season made those vines take off better, and it also meant the vines that have been fertilised went to sleep better. Okay. Now, I told you all that so we can sort of more or less get to this. The relationship on the left-hand side, it's just a schematic, more or less describes the response curve to most inputs. In this case, it might be vine nutrient status, really, which is the percent N, for want of another term. Really, when things are deficient, you put a little bit on and you get a fairly major effect, and then you get up to a point where it's marginal. You put a little 1 kilo on and nothing happens. It just stays the same. And then you keep pouring money into it, and you move along the adequate range until you get to, and then nothing's happening. And then until you get to high, and then it's excessive or toxic even. And that does happen. You don't get it. It's pretty hard to achieve that in Australia, but plenty of people have been trying. The point is, though, we really only want to be just at that marginal, maybe in the early parts of the adequate things. And we know what those adequate levels should be with petioles at flowering. Now, petioles at flowering are just a standard method of assessing vine nutrient status. There's nothing particularly magical about those petioles at flowering, opposite the basil bunch. It's just that everybody can recognise it. And that's what makes it an easily useful standard. When we talk about how many milligrams of nitrogen, which is another method of using those same petioles, you can extract the nitrate from those petioles, and you can get an instantaneous measure, if you like, of how much mineral N is in the petioles at that time. And we probably know a little bit more about what those might mean, particularly in relation. But the point I want to make in the next slide or two is that we shouldn't consider these in isolation. Now, the first reason we shouldn't consider these in isolation. First off, just remember we only know what adequate means here, 0.8 to about 1.1, and we think we know what deficient, marginal, adequate, and high mean in relation to nitrate. When we look at the amount of nitrate, so those nitrate ranges and the amount of percent N, the amount of nitrate that is present in the vine or in those petioles, and presumably also the leaves at any moment, is actually quite a small percentage of the amount of total N. And it's the total N, the total organic N, which is in those in those leaves and petioles, that is doing all the hard yards. It's doing all the work. It's in proteins. It's in various other things. It's running all the metabolism. It's doing all the good gear that should be making you money. The nitrate that's in there is essentially useless. And the reason it's useless is because it's not in the ammonium form, the amino form. So that's NH2. And it has to go through a process for that to happen. Now, when we talk about, you can have quite adequate levels of total N's, be in that 0.8 to 1.1 range, and have nitrate N at 0. And it causes a great deal of consternation. It doesn't need to. If it was at 0 and you were quite adequate N, total N, all that means is that you hadn't put your fertigation system on yet, and you hadn't put any, you haven't tried to work in any cover crops. That's all it means. So nitrate is a useful indicator of that any fertilizer that you're doing right now is actually working. And you might, and given the scheme of when uptake really kicks in that I mentioned earlier, you would have turned on your fertigation system about 2 or 3 weeks before flowering, because you were trying to, because that's the period when you're starting to get that uptake. Okay. You would have turned it on. So it would have been continuous. So the amount of nitrate that you would find in your thing would be probably reasonably low. It could be in that 340 to, 500 range, provided you weren't trying to overdo it. So the point I would like to make is that we have percent N, and that's a good measure. It's because that's where most of the business is actually happening, and we have nitrate N, which is a useful index to assess the efficacy of our fertigation programs. Okay. Now, occasionally in Australia, there was some a couple of Aussies who, no, people from South Australia, I mean, from Sardi, who who worked on a program a few years ago looking at, you know, a bunch disorder caused by poor fruit set. And they were having nitrate N levels of around about 2,500. Huge, absolutely huge. And you look at that lower number there, you know, it's almost 20% of the total amount of nitrogen in the thing. The problem was that the nitrate was in there, but it wasn't being converted to nitrate, to amino nitrogen, because it was one of the a significant cofactor in that process was missing, and that was molybdenum. It's a pretty rare thing, but these sort of things happen. So that the nitrate is a reasonable indicator of the efficiency of your fertigation system, whether you're not, you know, you're not rinsing it past, or you're not whatever, and also an internal physiological problem. So anything above 1,200 should be starting to cause a little bit of concern. When it gets up to 2,500, man, it's time to have a good look at yourself. Okay, now we have around here tried to incorporate vigor into to assess our vine our nitrogen management programs. And so all we've tried to do here is just use internode length. And there was a period in the dried fruit industry, for example, where internodes were, you know, anything up to 200 millimeters long. It was huge. We know obviously we didn't want that. We wanted internodes that were probably about as wide as your fist, more or less, because that equates to more buds per meter of bearer. And when buds are 200 millimeters apart, and you're trying to restrict bearer number or bearer length, or whatever it might be, or your machine pruning or something, you get fewer buds. So that's not a good thing. So what we tried to come up with was just some sort of interpretive table, and which I've tried to present here. And if we just confine ourselves to up the top to a short internode length, and then the percent N, which was down less than 0.8, we'd probably be saying, well, hang on, something's wrong here, buddy. Might need to improve the amount of nitrogen in the more nitrogen supply is probably limiting, but equally water is equally limiting. We're probably not watering them quite enough. Or go diagonally across all the way to long internodes and total nitrogen levels being above 1.15. We've been a little bit generous. You know, too much N and too much water. And in some senses, in some periods, we were starting to believe that when we watered them excessively, we thought we needed to put more N in to keep them going. And then when we put more N in, they grew more. We thought we needed to put in more water. And it became something of a forward-feeding reaction. And vigor just got out of control. And that was particularly on Ramsay rootstock, for example. So, yeah. So this is you know, it's not I wouldn't get too you know, hardcore about this, but a reasonable indicator of vigor, and using the number one eyeball to see how they're going. Look at those, and looking at those internode lengths and the petioles. You know, they're pretty. Yeah, there are relatively simple ways of adding a lot of value to that total N figure, as well as, of course, that nitrate figure on top of that. Okay. Now, try and summarize a lot of this. We've spoken about, this is a fairly simplified version of the earlier diagram I chucked up. If we just concentrate on the left-hand side, so that's nitrogen fertilizers. Now, what I've tried to indicate here, that it's really needs and placement. And by needs, I mean we're assessing how well fed those vines are, and then we're placing suitable amounts of fertilizer in the root zone. And I'll talk about the temporal nature of that in a minute, but matching needs, getting that right, and then placement. So we're putting it where the roots can get it. So chucking it on the surface and hoping they'll just come up and grab it and take it back down, it's not going to cut the mustard. Getting it into the root zone is what is really required. Leaving it in the root zone. Don't rinse it past. And that's the idea of that that red arrow going off to the left, avoiding oversupplying water beyond the readily available water level. And we've all done soil surveys. We know what the readily available water levels are. The measurement's not perfect, but gee, it's a heck of a lot better than just lagging it. Oh, well, just make it up as you go along. So avoiding rinsing it past. And the needs bit is met a little bit by that phenology thing there, when those vine roots are really starting to take off and they're starting to look for it. So, and when we want to manipulate particular things, initiation, shoot growth, and blah, blah, blah. In well-aerated soils, and also avoiding waterlogging, so to avoid that loss of nitrous oxides. Nitrous oxides, the production of nitrous oxides are encouraged under waterlogging conditions. So that means keeping an eye on the weather and not supplying nitrogen when there's likely to be a big downpour and might be waterlogging. Because, well, two things. It'll push what nitrate is in there down below the root zone because it's beyond the readily available water levels. And it will also cause localized waterlogging, which represents an economic loss, basically, just money up into the atmosphere. And we go across to ammonium fertilizers. Again, it's the same sort of deal. Needs and placement, and same with urea, getting it into that ammonium form. Avoiding high relative humidity. So again, avoiding when there's going to be a rainfall event. It depends on the soil type, though. Acid soils are more likely to be this way inclined to lose ammonium as ammonia. So when it comes to the ammonium fertilizer, it's against the same things. Phenology. It's just looking at putting these things on when the vine is looking for it. With organic nitrogen, it's all those, and here I've included, the cover crop residues. It's all about composition. So that C to N ratio, which more or less describes the way that these cover crops will behave. The higher the C to N ratio, the less likely it is that, more likely it'll take a long time for that stuff to break down. And the lower it is, conversely, of course, the lower it is, it's more likely it's going to be moving on pretty quickly, being mineralized pretty quickly, which may not coincide with when those vine roots are looking for it. It's also a question of incorporation. I don't know if any of you have gone past where people have thrown on composted cow manure. If it's not incorporated, that stench hangs around for quite a long time. So there's volatile compounds being lost from that stuff when it's just sitting on the surface. When that material is incorporated, those losses are obviously significantly lower. Well, for a start, just the stench, the smell is much reduced, which can only tell us that volatile compounds aren't making it to the surface to the point where it's noticeable. Okay, so that slide I was also going to say something about the conversion of ammonium to nitrate. In well-aerated soils, that happens quite happily. But if we're wanting to slow that down, there in some production systems where the returns warranted, nitrification inhibitors are becoming more common. Their use in viticulture is probably, there's just much less experience. I really can't say too much more about that. We haven't seen them being used to the extent they've been used in other perennial production systems. Mind you, they've been used in broadacre systems, so where things like fertigation just simply aren't possible. And they seem to have worked out the economics. So maybe there's something in that, but I don't think I could comment on much further on that. Now, across the top, when we spoke about the Haber-Bosch process and the philosophical hurdle that many people had with using that urea or ammonium fertilizers that resulted from that particular process. Maybe in the future, as renewable low-cost energy becomes more available, and the whole energy, fossil fuel usage problem becomes less significant, maybe some of that, a little bit more rationality may ensue from that, and people may be able to overcome that philosophical hurdle in their minds that they shouldn't be using these particular materials. But again, that's up to them. But renewable energy is becoming more widespread, and the cost is coming down as we all put on solar panels and various other things. So yeah, maybe that's going to be quite a useful development for the future. Okay, now just quickly sum up. Matching supply with demand. It's key. Vine phenology, vigor, and nutrient status. Slow release fertilizer. Okay, some may have an issue. Some may be, the organic producers may think this is the way to go. Organic versus synthetic, of course, as I said, there's a bit of a hurdle there. Nitrification inhibitors may start to play a bit of a role, but I wouldn't like to overstate that too much. I think the biggest thing is just avoiding supply being much, much, much greater in both time and space. The movers and groovers call them spatial and temporal, but time and space. When we put it on and where we put it in the root zone and when the roots are looking for it. So that raises the issue about micro-dosing, fertigation, and then intra root zone placement, just getting it down there in that part of the root zone where most of the roots are. And you can tell where that is because you can tell where most of the soil moisture is being extracted from. Okay. And that's, it depends on the soil, depends how deep the soil is, depends on the root stock, of course. But that's, the soil moisture monitoring systems are available now to tell us that. And it stands to reason that there may be roots all over the place, but a lot of them are structural. But it stands to reason that wherever most of the water is being extracted, that's an ideal spot to get that material into. Foliar, I've put in there a bit of a question mark, as I said, much less experience more generally with that than with other, particularly in the wine grape industry, than with other more conventional ways of doing things. But as I said, the table grape industry, they swear on that. Citrus, for example, you can supply probably pretty well most of the citrus trees' needs, which is via foliar. So some plants are more suited to it, certainly, than others, but much less experience in the wine grape industry. Avoiding leaching and waterlogging. Weather predictions are becoming far more reliable. Predictions further out, out a fortnight or so, are becoming, pretty good. They're not perfect. It's a pretty, pretty chaotic system, the atmosphere. But they're becoming pretty good. And the ability to incorporate those predictions into management decisions, I think is, a good thing. Now, I don't want to dwell on this too much. It'll be in the production, in the video that you are able to download. But there are some publications that I think could be quite useful to you, should you so choose. And some of them are, a bit more accessible than others, a bit more readable than others. But yeah, I think they were quite useful to follow up. Well, yeah. Now, quickly, I just want to make a couple of quick comments about carbon. There's a fair amount of carbon in our, well, we think there's a fair amount of carbon in our soils. There's actually very little. We think there's, when we tend to get a bit excited when people start talking about carbon farming and that kind of thing. I think it bears thinking about, certainly, but and it doesn't stop people acting collectively, but really there's not a lot of organic matter in our soils anyway. Now, and I can't remember if any, I don't know if any of you can remember the amount of what they were talking about of tons of CO2 equivalents was going to be worth. But when the carbon tax was first spoken about quite some years ago, they were talking about a carbon price of about $20. When you look at the amount of organic matter in our soils, and you start doing the numbers, this is up here where our soils are lower in organic matter than many other places around the country and many other wine grape producing regions. There's only 66 tonnes of about CO2 equivalents in the organic carbon. 66 tonnes. It's not a huge amount. So $20 a tonne. It's not. We're not going. No one's going to be going buying a new Toyota based on that. And you've got to keep that and keep it going upwards, and guarantee that for quite some time. When we look at the amount of carbon that's actually in a vine, the numbers are obviously vastly less. Now we know these are reasonable numbers because we've dug vines up and quantified all these. It's a dreadful thing to do. So if you want to see if I'm telling lies or something, go and dig one up, and I can assure you you'll accept this pretty quickly shortly thereafter. We've done all this with the aim of quantifying the pools that are in vines, and really there's not a lot in it. Across a hectare, tons of carbon per hectare and then tons of CO2 equivalents per hectare, there's only 24 tons of CO2 equivalents in the perennial parts. Now that's in an established vineyard, and you've got to take that. You've got to be able to guarantee that out for quite some time. That's pretty hard. So anyway, just food for thought. If you don't remember anything else, just remember that there's probably about 25, 35 tons at the height of summer in those vines. It's not a lot. Anyway, okay. Thanks for listening. Good luck with it all. And if you have any burning questions and questions, I'll try and answer them.
Oh gosh, I've been talking and I have had myself on mute. That's very professional of me, isn't it? So you would have seen the poll that popped up. So thank you for filling that out. And Michael, thank you for your fabulous presentation. We have a heap of questions. So the first one is, what is more valuable for carbon and nitrogen, putting straw mulch under vine or in mid-rows?
Well, I'll draw your attention back to a statement I made that there's a lot of other good reasons to use all that sort of material. If we're putting it under the vines, remember there's a nitrogen drawdown, but the benefit of the nitrogen under the vines is that it will suppress evaporation a great deal, and it will also suppress summer weeds. But equally, that goes. The amount of nitrogen, having all that straw mulch between the rows, which is mostly dry most of the time, would mean that soil is cooler, and vine roots will be tending to explore that more. There may not necessarily be all that much water over there for them to get, though. So it's sort of, what do you? It's if you're trying to suppress weeds, it's a good way to go, because well, I don't know if anybody's aware of it, but glyphosate's is in the firing line more recently. So stopping the use of or reducing the use of those chemicals is a good thing. It's really a question of what you're trying to achieve. I don't think you can say something's more valuable. It's really what you would like to achieve. It's also important to realise how much material you actually need to have an impact. And I can recall being over in eastern Victoria where they piled that stuff up, and in very short order, it had collapsed down to the point where it compressed down to the point where it produced a real blanket, but they needed to put on a large amount of material, and that stuff's not always available. Throwing that stuff on using a side throwing slasher or mower is probably a better way to go, but the particle size is so small that it probably breaks down reasonably quickly. Yeah. I don't think you can give an unequivocal answer, really. Yeah.
All right. That was good food for thought, though. So Jeff has written a very common PPL result is adequate to high total nitrogen combined with a very deficient nitrate. Now, you did talk about this, but can you explain again how you would interpret this?
When vines take up nitrate, it's transported, it's either converted in the roots or it's converted up in the top to amino N, and that's what is the biggest component of total N. Now, when vines take up nitrate the previous season, it doesn't sit there the whole time being nitrate. It is metabolized to be amino N, which then enters all that cycle. So that's the perennial parts and so forth, and comes out ready to rock and roll in the spring. So the total N levels are probably quite adequate, but if nitrate levels are low, it means that either A, the fertilizer program hasn't started, okay, or it's a little bit inadequate. That's all it means. It doesn't mean those vines are actually nitrogen deficient.
Okay. Great, thank you. And Patricia says, really interesting, Michael. We aim for about 100 millimeters for internode length. So yeah, I really like the table that you showed where you assess the needs of the vine for nitrogen based on vigor combined with the nitrogen analysis results. So yeah, I'd really encourage people to jump on and have a look at that when you receive the recording. It's just a really good tool to and really practical, easy to use. So thanks for sharing that. Lucy Etheridge says, after a couple of very soggy seasons and already high water table localized water logging, heading into this growing season, what are your suggestions for optimum nitrogen applications for good, efficient uptake?
In those conditions where it's waterlogged, apart from it's just being difficult to do a whole stack of viticultural practices such as spraying and so forth, it's really a question of hanging off, because it's likely that anything that's chucked on is going to be lost reasonably quickly. So there seems little point. The only thing, it's down in the Yarra Valley. It's a bloody swamp anyway. So it's awfully hard to. It's wet and conditions down there are pretty difficult. The one thing to consider is just sowing something that's going to take off quickly and just pump all that water out. And something like oats. The soils will be starting to warm up a bit, and sowing something that germinates quickly and gets it out, and it's easy to kill, it probably should be considered. Now, all the ryegrass and the other things in that particular where Lucy is, in fact, I think I know that vineyard, where Lucy is, the ryegrass and all that, they don't actually chew up all that much water. So expecting them to be able to pump all that out in a reasonable time frame is probably a little bit, unrealistic. So getting something in there that'll actually just reduce the water would probably be a way to go before even considering putting anything on. There's likely, and I wouldn't sow those cover crops. I wouldn't sow that interrow area with any fertilizer at all, nitrogen fertilizer at all, because it's just going to be, good money after bad, basically. Yeah.
Would you use a foliar fertilizer application?
For that cover crop?
For the vineyard.
In that particular situation, you'd probably want to investigate it, yeah. And it's not that hard to do to make a comparison either. You just you can leave rows unsprayed and just look at it. The leaf greenness will let you know whether it's working or not. Yeah, but the temptation is that if a little bit is good, a whole lot must be fantastic. Don't just I can't remember, and I'll follow up what sort of levels the table grape guys use. And remember, they're on another they're in a different production system, and that could be used as a basis. But for wine grape production, I'd be backing off a bit. Yeah.
Thank you. So can nitrogen or other nutrients, can fertigation application help reduce leaf loss if applied before a heat wave event?
I might have to take that question on notice, I think.
Okay.
I think I know the answer, but I'm just not quite certain. It's likely, although I would say that if the best thing about an oncoming, best thing to do for an oncoming heat event, and as I said, weather predictions are getting pretty good, is making sure that the vines are pretty happy when they go into it. We do know that during those heat events, they just stop. It's too hot. They don't produce anything. They don't transpire. So it's not like they're under enormous evaporative demand. What the thing that they are under is enormous oxidative stress. All that energy has to be dissipated by something. And normally those leaves are able to do that. They have a certain amount of cooling by actually pumping or letting water evaporate out through the stomates. And that there's a certain amount of cooling result there. But when you know one of those things is going in, making sure that the soil is at field capacity is a good thing. The other thing depends on what the conditions are like, but microclimate manipulation. In some cases, particularly on our vineyard, when we were grape producers, we had an old overhead irrigation system. We'd just turn on the sprays for an hour at lunchtime, cooled the whole place down. The table grape guys are onto this as well. They have cooling things, and in fact, I think there was a bit of work in South Australia by Mike McCarthy looking at vineyard manipulation. So keeping the vineyard floor wet, so evaporation causes cooling, and making sure that those the soil is topped up on the way as they go into that event, and if at all possible, altering that microclimate at the canopy level as well, if possible. Yeah.
Great answer, thank you. So David Yates, if adding composted chicken manure, how long would it take to get to the root zone? So if you're adding it into the surface, how long would it take to get down?
It will take a long time. A lot of chicken manure there's a lot of phosphorus in chicken manure, and it doesn't move all that quickly. There's a lot of calcium in chicken manure as well, and ditto, it doesn't move all that quickly. The nitrogen that's in it is generally in the organic form, so it's going to move slowly, if at all. It's probably much much faster. But I don't know. I think the best bet is incorporating it. Where that material is used locally, I don't know if anybody can remember what the old hydraulic silly plows look like, but they open up a delve on the side of the vine, throw it in, cover it up straight away. That's quite an acceptable and efficient practice.
Okay, great. Thank you. How efficient is calcium nitrate for uptake compared to urea?
They're both the same. If you're going to be using calcium nitrate, it's a better material in the sense that you overcome that. The longer you negate to some degree the longer term effect of excess cation over anion uptake, and that leads to soil acidification. So that's, and by excess cation we mean there's far more calcium, magnesium, blah blah blah, removed than there is, say, sulfate, phosphate, nitrate from the soil. And when that happens, this is just a natural phenomenon, and in the normal course of events it didn't matter because most things just fell to the ground and the net effect was zero. But where we're removing a crop, the grapes, we are contributing in the longer term to soil acidification. And that process will happen on a decadal scale, not this week, a decades. And so it's really, the coming generation that will need to cope with that. Management of that problem is difficult and expensive. Yeah. So there's really there's no difference in their relative efficiency as far as uptake goes.
Yeah. Okay, but the long-term impact is that acidification.
Yep.
Okay.
Yep.
So can foliar applications of nitrogen benefit from fulvic acid use?
Good question. I don't know. I've heard how many people tell me, and no one's produced any results for me at all. Particularly people who who operate in that space. I don't doubt the sincerity to which they hold those views, but I'd like to see some data. Yeah, if it works, great, just great.
Right, thank you very much. What a great presentation. I really enjoyed the session today, Michael. Thank you. Yeah, that's a lot of years of experience that you've shared with us during this webinar. So thank you very much. So our next webinar is on Thursday, the 21st of September, and Dr. Anthony Borderman from the AWRI is going to present on understanding Brettanomyces and its adaptation to control measures. So if you'd like to register for that, jump on the AWRI website and register. We would also like to make you aware of the Advanced Viticulture course that we are running in October. We have 2 courses running, one starting on the 16th of October, the other one starting on the 30th. We have only a few spots left on these courses, so it's 4 days. We jam a huge amount of content into those 4 days. We get to visit many, many vineyards in McLaren Vale, the Barossa, Riverland and Adelaide Hills this year. You get to hear from experts in the field, and you get to see some of these practices, techniques, tools in action in the vineyard. So if you are keen, please jump on. There's a QR code there, so you can use that to register. As I said, we've only got a few left, so be quick. And thank you everyone for all the great questions. It's always great to get the interaction with the audience, and thank you again, Michael, for your time. We look forward to seeing everyone at the next AWRI webinar. Thank you.
All right. Bye.